Hello, so this is Gary Roble, and I just wanted to go ahead and give you some video presentations on at least the first few chapters here to get you a little bit started in case you haven't quite got your textbook yet, or if you just got your textbook and haven't had much of a chance to dig through it yet. There will be a test coming up, probably the beginning of next week, Monday or so, Monday, Tuesday. I'll get you more information on that, but I did want to get some videos posted ahead of time. I don't know that I'll get them all done for the first 6 chapters, but I should get through at least 1 through 4 probably between now and Thursday or Friday of this week, so you'll have something to refer to. Okay. Anyway, moving along, I'm going to kind of highlight some of this stuff, and by no means does this make up for everything that's in the chapter in the book. I'll try to highlight a lot of the important information, but there's still quite a bit more that's in the textbook, so please read that and take a look at that closely. And there's on the website, of course, there's some student guides and practice questions and stuff that will help you with the process. So without further ado, let's go ahead and get started here in Chapter 1, A Perspective on Human Genetics. So genetics is the scientific study of heredity. Geneticists study how traits and diseases are passed from one generation to the next. And understanding what genes are, how they are passed from one generation to the next, and how they work is essential to understanding life. A gene is the fundamental unit of heredity. It's made up of DNA, and DNA is comprised of a polymer, which is a linked string of chemical subunits called nucleotides. Now, there's 4 different nucleotides in DNA. They are adenine, thymine, guanine, and cytosine, represented with A for adenine, T for thymine, G for guanine, and C for cytosine. Combinations of these 4 nucleotides define which amino acids will be used to make specific proteins in the cell. Genes are composed of sequences of these nucleotides contained on a double-stranded helical DNA molecule. The DNA molecule is made up of a base and a phosphate sugar, and we call the base, the phosphate, and the sugar combined the nucleotide. And so there's 4 nucleotides, A, G, T, and C, and DNA. There's a little bit of a larger picture of it. You can see the units. So you'll notice here that A seems to always code with T, and G seems to always code with C. In DNA, that is true. And the DNA is actually showing here the strands unwound. And this is great for if you're going to go to replicate things. You can unzip it kind of like a zipper and make a copy, or utilize the existing strand as a template and make a copy of one of the strands. But generally, DNA tends to be wound up like this. And let's go ahead and continue on. Now, there's a 3-dimensional structure to a protein. When a gene is turned on, you'll find that there's information in the gene that can be decoded, and it makes a protein that folds into a 3-dimensional shape called a conformational shape. You'll often find genetics depicted in art and culture. Artists have portrayed individuals with genetic disorders in different times and different cultures, and people with inherited disorder appear in myths and legends of many cultures. Again, this course is really designed as a overview of genetics and medical genetics, and tying it into heredity as a scientific act, and how it impacts society. So you'll often see us kind of switch gears periodically from talking about science and then about how it actually relates and how it impacts us on a daily basis, and why we need to understand this information as a conscientious and intelligent voter. For a lot of these issues are going to be coming up, and many of these issues are not dealt with in the public domain adequately yet, so there's going to be significant opportunities for you all to vote on them in future years, as some of this stuff will start to appear in legislation, as some of it will need to be dealt with. So in this case here, we're just talking about genetic disorders in culture and art, but we'll certainly talk about some of the other more challenging and technically relevant things that will be impacting society here in the future. And so that's kind of what this course is all about, the impact and the cross-section of science and in society. Okay, traits. Any observable property of an organism is a trait, and actions of genes, products, proteins in particular, produce visible traits such as eye color and hair color. Now, how are traits transmitted from parents to offspring? Well, the first work was done by Gregor Mendel, who was an Augustinian monk in Austria. He worked on pea plants in the mid 1800s and provided the foundations for the science of genetics. You probably remember him from previous biology classes dealing with Mendelian squares or Punnett squares. Now, Mendel's experiments showed that genes are passed from parents to offspring. Each parent carries two genes that control a trait, and each parent contributes one copy from each pair. Pairs of genes separate from each other during the formation of egg and sperm, which is called meiosis, and when an egg and sperm fuse during fertilization, genes from the mother and father become a new gene pair. Genes are contained on chromosomes. Chromosomes are found in the nucleus of human cells and other higher organisms. Meiosis separates chromosome pairs during formation of egg and sperm. Many different model organisms have been used, ranging from bacteria to plants to insects to humans. When we talk about transmission genetics, we're actually looking at the study of inheritance patterns and how traits are passed from generation to generation. We'll oftentimes do a pedigree analysis, which is a construction of family trees that are used to follow transmission of genetic traits in families and how it's passed on or how it's inherited from one generation to the next. A pedigree represents the inheritance of a trait through several generations of a family. So here you're seeing boxes representing males and circles representing females, and a horizontal line between the male and female, meaning that they were, I guess, consensually interactive, and so the male and female here in section 1, the uppermost section here on this diagram, shows that this male and female mated and they produced 3 male offspring. You have 3 boxes and 1 female offspring, and that female obviously married married or had a relationship with a male that produced so let's see, 1, 2, 3, 4, 5, 6 male offspring and a female offspring. Now you can see in the second generation here you have 1 darkened black box. That means that that person got a particular trait in question, whatever one you're looking at. But the brothers and the sister did not. Now the sister, when she mated with the male in generation 2, and produced the 6 male offspring and 1 female offspring, you could see that 5 of the males were affected by this particular trait. 1 male was not, and 1 female was not. So that's kind of what a pedigree does. It allows us to kind of see how traits get passed from one generation to the next and through whom they get passed. Cytogenetics. Cyto means cell. Genetics, of course, the study and dissection of how information gets passed from one generation to the next. So when you put it together, you get the study of the organization and arrangement of genes on a chromosome and the study of chromosome number and structure. A karyotype is a complete set of chromosomes from a cell that has been photographed during cell division and arranged by size and shape in a standard order. So here's a karyotype. It arranges the chromosomes in a standard format so they can be evaluated for abnormalities. As you can see here, you'll generally notice that the biggest ones, the longest ones, start at number 1. A little bit smaller is number 2, a little bit smaller number 3, 4, 5, so on and so forth, all the way down to 22. And then you have your X and Y chromosome here. In that case, you have X and Y, meaning that this is a male individual. If you had XX, it would be a female. There's no possibility of having a Y. XY and XX are the 2 options for male and female. Now, you have 22 sets of autosomes. Those are the cells throughout the regular chromosomes throughout the body, and then you have the 2 sex chromosomes, which is your 23rd pair. Okay, how do scientists study genes? Molecular genetics is one way, the study of genetic events at the molecular level. This is particularly looking at the identification, isolation, and analysis of specific genes. And then population genetics, the study of inherited variation in populations of individuals, forces such as environment that result in changing gene frequencies over generations. Genetics and basic and applied research. Basic research is generally doing research just for the knowledge of it, for the sake of doing it, to get some general understanding of a concept, not necessarily for any particular reason. Excuse me. Applied research is when you do research with intent or for a particular reason to show cause and effect, for instance, or show that there's a potential outcome that can be derived from a research project or an assignment. So let's take a look at a couple of these here. Recombinant DNA technology. These are techniques whereby DNA fragments are linked to self-replicating vectors, which are then replicated in a host cell, often like a bacteria. You have genetically modified organisms that fall in this category, as recombinant DNA technology can make genetically modified organisms, and you can carry and express genes from another species in utilizing recombinant DNA technology. Now, a clone, what you'll find there is you have genetically identical molecules, cells, or organisms, all derived from a single source or parent. This is often utilized in gene therapy, and normal genes are transported or can be transplanted into humans with defective copies to treat genetic diseases. How do we apply this to biotechnologies? Well, in medicine, you produce vaccines, like they're doing right now with COVID, and customized proteins for treating disease. Other segments like agriculture, we can utilize it to increase crop yields, lower fat content in foods, and produce disease-resistant crops. And genetics has also affected social policy and law. Again, here's that intersection of society and science. And one of the things that was an issue at some point in the past, early mid 1900s, was eugenics. And they utilized or misutilized genetics as a social policy. Eugenics was the attempt to improve the human species by selective breeding. So there was a series of legislation that was passed early and mid 1900s that attempted to improve the population by preventing immigration and the introduction of less desirable traits. There were laws that restricted reproductive rights and requiring certain individuals to be sterilized if they were instituted or were instituted. This was used by Nazis in Germany to promote mass killings of individuals believed to be genetically inferior and ultimately establish genocide. So eugenics was not, ironically, there's still some eugenic laws on the books, I think, in certain states here in the U.S., but by and far, I think for the most part, they've largely been overturned. But this was policy back in the early 1900s that was actually set in stone by the U.S. Supreme Court and everything. So this was something that was a very serious concern for a lot of years, and over time these things had been mostly overturned. But something very interesting piece of U.S. history to take a look at if you're unfamiliar with it. In the early part of the 20th century, eugenics exhibits at fairs and similar events were used to educate the public about the proposed benefits of eugenics policies. Carrie Buck and more than 60,000 others in the United States were sterilized for appearing genetically, intellectually, or morally inferior. A pedigree of the Buck family made up the Virginia colony for the epileptic and feeble-minded. You can see this listed here. If you look there under the legend, you'll see F for feeble-minded, name unknown. And from what I understand about Carrie Buck, she wasn't feeble-minded, or at least not in any kind of hereditary way, nor did she have any illegitimate children. I think that was a cause of concern that she had been promiscuous or had illegitimate children, if I remember the story correctly. But none of that was true and stuff. So they were just utilizing this to make a particular case. There's more about it in the textbook, so please take a look at those articles and stories, and they're quite interesting. Hitler and the Nazi Party used eugenics as an excuse to kill handicapped children and adults, in addition to Jews, gypsies, communists, homosexuals, and political opponents of the government. The close association between eugenics and Nazi Germany soon led to the decline of the eugenics movement in the United States. So that wasn't until like the mid to late 1930s. What impact is genetics having? Well, new technologies permit the identification of and determination of function of all 25,000 human genes. Genes associated with hundreds of genetic diseases have been cloned and used to develop genetic tests and new treatments. In vitro fertilization genetic testing help families make informed decisions concerning family planning. Stem cell research offers hope for treating many diseases, and the use of recombinant DNA technology or biotechnology allows the production of novel plants and animals, genetically modified organisms. So here you see transgenic corn, which has been genetically modified to be resistant to herbicides, which are used to kill weeds. So the plant's resistant to the herbicide, but the weeds obviously are not, so they die off, and you have a nice clean crop of transgenic corn. Began in 1990 with the aim of determining the entire sequence of the 3.2 billion nucleotides of the human genome was the Human Genome Project. This was quite an undertaking, and through the use or development of computers and PCs at this point in time, in the early 1990s, the Human Genome Project was actually able to complete a little bit early, if I'm correct. I believe I am. Analysis of SNPs, or single nucleotide polymorphisms, which are changes, show differences among individuals at the molecular level. For instance, if you look here at row 1, person 1, haplotype 1, you can see there, if you look at the first letter C, the red C, the single nucleotide polymorphism there. Normally, if you look at person 2, 3, 4, there'd be a G there, but in this individual, there's a C there. So there's something that's unusual or irregular there. Person 2, you notice that there's an A at the third SNP for haplotype 2. Normally there should be a T there, but in this particular individual, person 2, there's an A there. In row 3, person 3, the haplotype 3, there's a SNP, the second SNP. You have a C where normally there'd be a G. But as you can see in person 4, sometimes there's even a T there in that single nucleotide polymorphism at that particular location. You could have G, C, or T. Looks like most commonly it's going to be G, and they're listing C and T as unusual events for that particular gene sequence right there. So that fourth SNP2, that's this person 4 actually has a secondary one in the thymine column for haplotype 4. You can see there's an adenine there instead of thymine, which the haplotypes 1, 2, and 3 generally have. A little larger picture of it. And new genetic technologies are gene chips that can carry all the known human genes, and we can use that to help diagnose genetic disorders. Genes associated with hundreds of genetic diseases have been cloned and are used to develop genetic tests such as cystic fibrosis, sickle cell anemia, muscular dystrophy, and PKU or fetal ketonuria. In vitro fertilization embryos at this stage can be analyzed for genetic disorders before implantation, which is kind of a nice ability to be able to tell this stuff. There's a lot of testing during pregnancies nowadays, and some things, severe genetic disorders, might be able to be determined at a very early stage. Animal cloning, Dolly was the first cloned mammal. You may have heard about her in the history books or whatever. I forget the year off the top of my head, but it's probably been 20 years or close to 20 years at this point in time since Dolly's been cloned. I don't think she's with us anymore. I think she's passed on quite a number of years ago. But anyway, she was a cloned mammal and the very first cloned mammal that we know of. So keep in mind that genetic technology has developed faster than the legal and social consensus about the use of genetic information. So as you'll see throughout this course, you're going to learn some technical details about genetics and how it works fundamentally. So you'll be able to discuss this stuff in an intelligent fashion when it comes to voting on some of these issues as they come to bear on us here in the future. Okay, what choices do we make? Well, we are now applying genetic knowledge in ways unforeseen just a few years ago. With the completion of the Human Genome Project, new methods of genetic testing, and the ability to transfer genes, we are faced with many personal and social decisions. Ethical use of genetic information and biotechnology requires participation by a broad cross-section of society, and we can make informed personal decisions and formulate public policy only if we have a knowledge of genetics and how genetics is used in biotechnology. And that's the end of Chapter 1. Thank you for your time.